Good morning, everyone, and welcome. I am delighted to have you here today. My name is Dr. S.K. Singh, and I hold a PhD in this field. Today, we will be exploring the stability of pharmaceuticals excipients. Over the course of this presentation, we will discuss what excipients are, why their stability is crucial, and the factors that influence their stability in pharmaceutical formulations. I'm excited to share this knowledge with you, so let's get started. Let's start by looking at the purpose of this document. Since excipients are different from active pharmaceutical ingredients, or APIs, and medicinal products, this guide provides clear guidance on how to develop supporting studies specifically for excipients. It also explains how this information can be shared with users and regulators, ensuring transparency and safety. Moving on to the pharmaceutical end-user perspective. If an excipient is stored properly, that is, in an unopened container at the recommended storage conditions, it will continue to meet the manufacturer's specifications or the requirements set out in the monograph, up to its shelf life. This highlights the importance of proper storage to maintain quality. Now, onto the principles of this guide. It's designed to be applicable internationally, so it aims to be relevant across different regions and regulatory environments. Because of that, it doesn't specify legal requirements or cover every possible characteristic of every excipient. While the guide recognizes that the ICH-Q1A and WHO guidelines are tailored for drug substances and products, it's important to note that their principles can still be useful for excipients, especially when establishing stability programs. For example, even if an excipient manufacturer doesn't perform stability testing exactly as the pharmaceutical industry does, they can still apply similar principles to ensure product stability and quality. Overall, the goal is to provide a solid, harmonized basis for excipient stability, supporting consistent quality across the industry. Let's now look at some general information about excipient stability studies. The main goal here is to gather evidence that the excipient will continue to meet the manufacturer's specifications throughout its shelf life. This means we need to identify the recommended storage conditions while the product is in its sealed commercial packaging. Additionally, it's important to determine how long the excipient can be stored, what we call the shelf life, and to establish the specific shelf life claims we can make. It's also crucial to understand that once the package is opened, any stability issues that arise are the responsibility of the user or anyone repackaging the product. So, stability testing focuses on the product in its original, sealed form. The document we're referencing also describes a suitable program for developing this stability data, ensuring that all necessary factors are considered. Now, let's move on to how excipients are classified based on their stability. As you might expect, not all excipients behave the same way. Some are more stable than others. To facilitate clear communication between manufacturers and users, we categorize their stability into broad groups based on how they perform in their commercial packaging. These classifications are determined using available stability data, which can come from internal sources or external studies. The classification helps guide how we approach storage, handling, and further testing. Finally, as we go deeper into this topic, we'll see that additional requirements, such as documentation and specific study protocols, are addressed in later sections. This structured approach helps ensure we maintain quality and safety throughout the product's life cycle. Let's start by looking at the category of very stable excipients. These are the ones that have demonstrated a retest or reevaluation interval, or expiration date, of 60 months or more within their specified commercial packaging. In other words, they tend to maintain their stability over a long period, which is quite advantageous. It's also important to note that their stability isn't usually affected by changes in the manufacturing process, which adds a layer of robustness. However, we should keep in mind that packaging can sometimes be a limiting factor for stability. If the packaging doesn't protect the excipient well, it could shorten its effective stability period. Moving on to stable excipients, these have a demonstrated stability of at least 24 months, but less than 60 months. They generally show a shorter stability window compared to the very stable ones. An important point here is that these excipients tend to be more sensitive to changes, whether that's in how they're manufactured or the type of packaging used, than the very stable group. So when working with these, we need to be a bit more cautious about process modifications or packaging updates. Finally, limited stability excipients are those with a stability period of less than 24 months. These are more at risk if there are any changes to their manufacturing process, raw materials, or even environmental factors. Because their stability window is shorter, any deviation or variation can potentially cause them to no longer meet product specifications. So, for these excipients, close monitoring and strict control are especially critical. In summary, understanding these stability categories helps us manage our excipients better, ensuring the quality and efficacy of our products over their intended shelf life. It all ties back to our overall goal of maintaining product integrity throughout its life cycle. When we're dealing with excipients that are new or chemically novel, it's crucial to gather stability data, especially when their stability profile hasn't been established yet. As development progresses, the classification of these excipients can change once more stability data becomes available, so staying flexible and attentive is key. Now, when it comes to modifications to existing products, we need to carefully evaluate any changes that might impact stability. These changes should be clearly defined, documented, and aligned with the principles of managing significant changes. Conducting a thorough risk assessment helps us determine whether a change is significant enough to require additional stability studies. Interestingly, if new packaging has been demonstrated to be equivalent or even better through other types of studies, like moisture vapor permeation or oxygen permeation tests, we might not need to conduct a new stability study. However, it's important that we document our justification for skipping this step. And depending on the results of our risk analysis, these stability studies can sometimes be conducted concurrently with the implementation of the change, helping us streamline the process without compromising safety or quality. Overall, the key is to carefully consider how new or modified excipients and packaging might affect stability, always using data and risk assessments to guide our decisions. This approach ensures we maintain product integrity throughout development and beyond. Now, let's talk about ongoing stability studies. These are important to ensure that excipients remain stable throughout their shelf life. For excipients that are very stable, once you've established a shelf life claim, there's typically no need to perform further ongoing stability studies. However, for stable excipients that are less robust, the frequency of these studies should be guided by a documented risk assessment and solid scientific justification. When it comes to limited stability excipients, ongoing stability studies are definitely recommended. In some cases, you might consider accelerated storage conditions, especially if the packaging closely mimics the final container and closure system. Overall, the key is to tailor your stability testing approach based on the stability profile of the excipient, ensuring product quality and compliance throughout its shelf life. Let's talk about stability information. It's important that any data related to the stability of excipients is available, especially if it supports the safe and effective use of a product. However, sometimes this data is proprietary, such as detailed compositional information, and in those cases, sharing it may require confidentiality agreements to protect the manufacturer's intellectual property. That said, a clear statement or reference to stability information should be included on the packaging to demonstrate the product's stability to users. Now, when it comes to classifying excipients based on their stability, there are a few key categories to consider. First, very stable excipients should have a summary report available upon request. This report would include relevant data like stability indicating characteristics, literature citations, analytical trending results, and monitored storage conditions. For excipients that are stable or have limited stability, a more comprehensive summary report supported by ongoing stability studies should be available. This report would include similar data points, ensuring users have the information they need. And finally, if there's not enough data to classify an excipient's stability, it's important to obtain additional information through dedicated stability studies. Overall, providing thorough, accessible stability data helps ensure safety, quality, and regulatory compliance throughout the product's life cycle. Let's take a closer look at the options available for designing an excipient stability study. Generally, options 1 and 2 are most commonly applicable in most situations. Starting with option 1, it involves utilizing existing historical data, which can include information found in scientific literature or internal reports. For example, we might look at data where an excipient was stored in a warehouse for a set period, then retested at the end of that period to confirm it still meets specifications. Alternatively, we could use documented evidence that the excipient retains its functionality after a certain time. Published data, whether from journal articles or internal sources, can also serve as valuable supporting information. Moving to option 2, this involves conducting a study using the actual commercial packaging and storage conditions. This is especially useful when those conditions are uncontrolled, such as variable humidity and temperature. Suitable data here might include retest results from batches that have been stored in real-world supply chain conditions over time. These real-world data points help us understand how the excipient performs under typical storage environments. Both options aim to provide reliable data on excipient stability, with option 1 being more about leveraging existing data, and option 2 focused on generating new data under actual or similar conditions. Most situations will likely benefit from one of these approaches, helping us ensure the excipient remains stable and effective throughout its shelf life. Option 3 can be used when we're working with novel or new excipients, especially if we don't have stability data in accordance with options 1 or 2. As noted in the IP-CPQG GMP guide, for excipients that are available in multiple grades, techniques like bracketing and matrixing studies, described in ICHQ-1D, can also be appropriate. It's important to have evidence of the time intervals between testing and storage, as well as clear information about the storage conditions used. We also need to define the commercial packaging and understand the general conditions under which the samples or containers are stored. To support this, a warehouse monitoring or mapping study, combined with facility maintenance records, can be very helpful, especially if we can demonstrate that the excipient remains stable across a wide range of storage conditions. The main goal of the stability study is to show that the excipient stays stable under the expected storage conditions. In many cases, excipients can be stored under uncontrolled warehouse conditions unless specific storage requirements are necessary for stability. If those conditions aren't known, establishing a warehouse monitoring program is essential. This involves monitoring the temperature in the storage area where the study and the commercial product will be kept, ideally for at least a year. This helps us understand the maximum and minimum temperature ranges caused by seasonal changes. Humidity also needs to be monitored, especially if the excipient is hygroscopic or sensitive to moisture, or if the packaging allows moisture transfer. In summary, thorough monitoring and understanding of storage conditions, temperature, humidity, packaging, are key to ensuring and demonstrating excipient stability during storage. When we talk about stability studies, it's essential that we describe them clearly in a detailed protocol. First, we need to define the objective of the study. What exactly are we trying to determine or confirm? Then, we move on to the scope of the study, which includes justifying why we've chosen certain materials and how many batches we plan to test. Typically, at least 3 batches should be included initially, especially when there's limited existing stability data or literature on the product. Next, we must carefully select and justify the grade of materials used, particularly in model studies or when producing multiple grades. It's important to consider risk assessments and justify how we include samples from different sites, ensuring they all use the same starting materials, formulation, equipment, processing, and packaging to keep the data consistent. When selecting batches, we should justify that they are representative of usual production, what we call typical batches, so the stability data is relevant to real-world manufacturing. Similarly, packaging choices need to be justified, especially when multiple packaging types are involved, by considering the worst-case scenario to ensure robustness. Storage conditions must be clearly defined, reflecting real-world conditions and any accelerated testing plans. The sampling plan should specify when and how samples are taken throughout the study. Stability test methods need to be detailed and validated, ensuring reliable results. Lastly, we define the acceptance criteria, what standards the product must meet at each testing point to be considered stable. Putting all these elements together in the stability protocol helps us create a comprehensive, transparent plan that supports product quality and compliance throughout its shelf life. Let's talk about the objective of the stability study. It's crucial to clearly state the purpose right from the start. Are we conducting this study to support a new excipient, to evaluate the impact of a change, or is it part of an ongoing stability confirmation? Clarifying this helps guide the entire process and ensures everyone understands the goal. Moving on to the scope, the protocol should specify exactly which excipient or grades of excipient are included in the study. This helps define the boundaries and focus of the study, making sure we're testing the right materials. When selecting samples, it's important to choose lots that represent a broad range of the product's normal variation, especially related to the stability indicating parameters. Ideally, we include at least 3 different batches for the initial stability study. However, if we don't have enough batches initially, starting with a single batch can be acceptable. For ongoing stability programs, a single batch is often sufficient. For new excipients, we can use developmental lots to gather initial stability data. But to truly confirm stability, it's recommended to use 3 or more commercial-scale lots. This provides more reliable and comprehensive information about the excipient's stability profile. In summary, defining the objective and scope clearly, selecting representative samples, and using multiple batches for validation are key to a robust stability protocol. These steps ensure our stability data is meaningful and supports the overall quality of the product. Let's start by discussing the selection and justification of representative batches. It's important that the batches we choose cover the full range of normal production, including filling and packaging processes. We should aim to include worst-case scenarios, especially in terms of key specification parameters, with a particular focus on stability indicating parameters. When possible, it's also advisable to manufacture these batches using different batches of critical raw materials. If that's not feasible, we need to document a clear written justification as part of our protocol. Now, moving on to the selection of packaging for stability studies. The key here is to treat both the container and closure system with the same level of care and consideration. Ideally, the packaging used in the stability studies should mirror the commercial packaging. This ensures our stability data accurately reflect real-world conditions. However, sometimes we might consider alternative packaging options. For example, using packaging that offers significantly less protection can lead to a shorter retest or reevaluation interval than what we'd expect with the commercial packaging. This approach can be useful to simulate a worst-case scenario, especially for protecting excipients during storage. But it's crucial that such choices are backed by solid scientific justification and demonstrated that the excipient's composition profile remains unchanged. An example of less protective packaging might be an unlined paper bag or a single-ply plastic bag, which offers minimal barrier protection. When selecting an alternative container, we should also consider surface area. For instance, choosing a smaller container with a larger surface area per kilogram of product can be relevant, especially if permeability is a concern. Lastly, if the alternative container is known to be more permeable or constructed from materials that allow more exchange, we need to factor that into our stability assessment. The goal is always to ensure that the stability study conditions are scientifically justified and representative of potential real-world scenarios. When it comes to stability studies for bulk shipments, such as barges, railcars, or tank cars, there are some unique challenges we need to consider. One key issue is the uncertainty around how long the excipient might stay in the bulk container. Because of this, designing an appropriate stability study can be tricky. However, We can often extrapolate data from the methods we do use, especially if we carefully consider the risk factors associated with different modes of transport and storage. Now, regarding storage conditions, it's important that we store our stability samples under the conditions specified in the protocol, or, when appropriate, under the conditions found in the manufacturer's warehouse. This ensures that the stability data is relevant to real-world scenarios. The stability study should cover the longest period that the excipient supplier guarantees the product will stay within specifications, using normal warehousing or the storage conditions specified. If we suspect, based on existing literature or data, that the excipient might degrade under normal warehouse conditions, then the study should be performed under more challenging conditions that test its stability. For example, if atmospheric moisture can hydrolyze the excipient, then the storage conditions should reflect that, say, storing below 50% relative humidity. In such cases, it's recommended to use controlled storage and stability conditions, along with a container and closure system that closely mimics the commercial packaging. This approach helps us ensure the stability data is robust and relevant. Sampling plan is a critical part of the study design. It's best conducted as a kinetics experiment, meaning that samples are taken at less frequent intervals as the excipient approaches its expiration or reevaluation interval. For example, you might test samples at 0, 3, 6, 9, 12, 24, and 36 months. The protocol should clearly specify the frequency of sampling, starting from the date of manufacture, which is considered time 0. This helps ensure consistent and reliable data collection over time. The main goal of the stability study is to confirm that the rate of change in the excipient's composition is slow enough so that it remains within specifications throughout its shelf life or reevaluation period. When removing aliquots for testing, it's important to do so in a way that doesn't affect the remaining excipient. This is especially crucial when the excipient is sensitive to exposure to the atmosphere. Opening the packaging can expose the bulk excipient to environmental factors, which may influence stability. Therefore, both during and after sampling, the aliquots should be protected from atmospheric exposure to maintain their integrity. If exposure during sampling could impact the excipient, it's advisable to consider using individual packaging that mimics the bulk package. This approach allows for sampling without compromising the rest of the stored excipient, facilitating accurate future stability testing. In summary, the sampling plan must be carefully designed to balance the need for representative data with the preservation of the remaining material's stability. Proper sampling techniques and packaging considerations are essential to achieve reliable and meaningful results in your stability study. Let's take a closer look at the methods used for stability testing. In the pharmaceutical industry, it's common to rely on assays as the main stability indicating test method. However, when it comes to excipients, this approach isn't always feasible or ideal. For instance, assays may not be suitable for monitoring excipient stability directly, especially if they can't accurately reflect changes or if the excipient's nature makes direct measurement difficult. In situations where we know the excipient may change over the reevaluation period, it's best to use validated or verified stability indicating tests that can clearly demonstrate such changes. Ideally, these tests should provide quantitative results, giving us precise information on the extent of any alterations. Stability of excipients can indeed be monitored through stability indicating assays, but if we can't measure the purity directly, we might need to look at other characteristics, like microbiological, physical, or chemical attributes, that can serve as indicators of stability. Now, a word of caution. When monitoring excipients through volatile decomposition products, things can get tricky. Because these products are volatile, there's a risk of losing measurable amounts during testing, which can lead to inaccurate assessments of stability. Lastly, it's important to remember that just confirming an excipient meets the specifications or passes an assay isn't enough. We also need to ensure that the overall composition profile remains consistent throughout storage, unless we have a justified reason to believe otherwise. This helps us ensure the excipient's stability and, ultimately, the quality of the final product. On this slide, we're focusing on what happens when results fall outside of our expected range, known as out of specification or out of trend. During a formal study, sometimes unexpected results can occur, and it's crucial to treat these findings carefully by following a formal out of specification procedure. To prepare for this, the study design should include extra samples beyond what's needed, so we have enough material to investigate if we encounter an OOS or OOT result, or if a sample is lost. The key point here is that an OOS or OOT result isn't automatically dismissed. It should only be discounted if there is a clear scientific reason. Ideally, the criteria for dismissing such results should be established before starting the study, so there's no ambiguity later on. However, we must recognize that sometimes it's not immediately obvious whether a result is truly out of trend until we analyze the data over time. This means investigations might not begin right away, but it's important to address these results systematically. Moving on to acceptance criteria, it's essential that our protocol clearly defines the limits for test results that support the product's shelf life or retest intervals. For each parameter, we need specific acceptance ranges or limits. Trend analysis plays a vital role here, as it helps us predict whether the excipient will continue to meet specifications throughout its intended lifespan. Sometimes, we might test beyond the basic specifications, such as looking at the composition profile, to gather more comprehensive data. And if we identify degradation products during stability studies, it's important to consider their safety and include specific criteria for those degradation products. Overall, these measures help ensure our stability testing is robust, scientifically sound, and aligned with product safety and efficacy throughout its life cycle. Let's take a closer look at the approval process. It's essential that the protocol clearly outlines how approval is obtained, including who is responsible for reviewing the data and drawing conclusions. In particular, it should specify where the responsibility lies for quality oversight, ensuring that there's accountability at every step. Moving on to stability reports, these are a critical part of the program. Throughout the process, the excipient supplier may issue interim reports to keep us updated on the stability data. Once all the data has been collected and evaluated against the acceptance criteria, a final report should be prepared. This final report should include a thorough statistical evaluation of the data, along with the conclusions that have been reached. If there are any deviations from the original stability protocol, those need to be listed and justified clearly. It's also important to note that statistical tools can be used to help determine retest periods, making the process more data-driven. The report itself should specify whether it's an interim or final report, and include key details like the excipient grade, the commercial packaging, and the recommended storage conditions. Test results should cover stability indicating characteristics and their acceptance criteria. If there are any deviations or investigations, those should be documented explicitly. Finally, the report must conclude with a clear determination of the shelf life claim and the retest or reevaluation interval, if applicable. And of course, the document needs to be formally approved, since this final step validates the entire process. All of these elements together ensure that our stability program is thorough, transparent, and compliant with regulatory expectations. Now, let's discuss the key points regarding excipient storage conditions and labeling. When excipients require specific storage conditions to maintain their quality throughout their shelf life, these conditions should be clearly stated on the product label or in other relevant documentation, such as the certificate of analysis. This ensures that anyone handling the excipient knows exactly how to store it properly to preserve its integrity. However, if no specific storage conditions are necessary for an excipient, then it's not required to include such information on the product label. Still, the manufacturer must have documented scientific justification that the storage conditions they recommend are effective in maintaining the excipient's quality up to the end of its shelf life, even when the packaging remains unopened. Additionally, if stability studies reveal particular conditions to avoid, like exposure to certain environmental factors, These should be explicitly specified on the label. This might include instructions such as protect from light or other protective measures. It's also important that the labeling for monographed excipients aligns with pharmacopoeial requirements, ensuring consistency and compliance with established standards. Overall, clear, accurate, and scientifically justified labeling is essential for maintaining the quality and safety of excipients throughout their shelf life. For further support, please don't hesitate to reach out. If you have any questions or need additional information, I'm here to help. You can contact us through the provided details. Thank you very much for your time and attention today. We truly appreciate your engagement and look forward to assisting you further.